Welcome to the neurology section of Falcon Physician Reviews. This will be the embryology section, where we will cover the embryology of the nervous system. In this image, what you'll see is a section of the embryo, and we're going to cover how the brain develops. At 3 weeks of embryologic development, the neural plate forms. The neural folds will come together to form the neural tube. Above the neural tube are patches of cells called the neural crest cells. Neural crest cells, you should remember, are not just patches of cells. They're actually 2 lines of cells that are going up and down the neural tube. In that way, they are very nicely situated to form the peripheral nervous system. Here's an image of the cross-section of the embryo showing the neural tube and where the neural crest cells are. The surface ectoderm will fuse across the midline, and if the neural tube does not properly close, that surface ectoderm will have trouble developing. And we'll talk later about different types of disorders that can come from that, such as spina bifida and anencephaly. The neural crest cells will migrate down and they'll form many different types of cells, and let's talk about what they form. Different types of growth factors will cause these neural crest cells to develop into different types of tissues, such as the sensory neuron, an autonomic neuron, Schwann cells, and melanocytes. The neural crest cells are the precursors to the peripheral nervous system. Therefore, they form the sensory and autonomic ganglia of cranial nerves 5, 7, 9, and 10. They also form the dorsal root ganglia and autonomic ganglia, such as the sympathetic chain. But in addition to that, they form non-neural tissues that are not quite as intuitive. These things you will have to just memorize through brute force memorization. The non-neural tissues include tissues such as melanocytes, cartilage, adrenal medulla, and facial bones. Let's talk about what the neural tube develops into. At the very front of the neural tube, where the brain is going to develop, there are neural tube dilations that occur. When they dilate, they form, at the very front, the prosencephalon, in the middle, the mesencephalon, and then behind, the rhombencephalon. So these would be referred to as the forebrain, the midbrain, and the hindbrain. But we're not done dividing yet. We still have to divide the prosencephalon into 2 more compartments and the rhombencephalon into 2 more compartments. The prosencephalon will further divide into the telencephalon. And I like to say that this is so far forward in the neural tube that you have to telephone to get to it. Okay, that's going to help you remember where that telencephalon is. The next section down is called the diencephalon. The mesencephalon remains the same, but the rhombencephalon divides further. The rhombencephalon will divide into the metencephalon and the myelencephalon. So there we're left with telencephalon, diencephalon, mesencephalon, metencephalon, and myelencephalon. And you may be wondering, why am I committing this to memory? This is such a pain and it doesn't really relate to my clinical practice. But indeed it will. There are terms that we use in neurology to refer to where, say, a hemorrhage may occur in the brain. So we may say this is a mesencephalic hemorrhage, or a perimesencephalic hemorrhage, or a diencephalic bleed. So it is important to remember these kind of terms. So how can we try to help remember these things? Well, we know that the prosencephalon forms the telencephalon and diencephalon. The rhombencephalon forms the metencephalon and myencephalon. And the mesencephalon remains the mesencephalon. You look at this and you say, well, why am I spending all my time trying to memorize the encephalon part? Well, it's all the same across each of the different components. So therefore, to memorize this most efficiently, you should be able to just drop the encephalon and think of tel, di, mes, met, and my. And there is a mnemonic to try to come up with this, but and I will share it with you now, but I don't know if it's going to be that helpful to you, but you can come up with your own. But you're going to need to remember that order: tel, die, mes, met, my. The way that I do it is I come up with a friend named Di. That makes sense. People named Diana may be called Di. You have to have a friend named My, and that's not so abnormal. Some people have, say, a friend that may be named My. But you also have to have a friend named Mez, and that's a little bit strange. But still, just for the sake of memorizing this, let's pretend that you have 3 friends: Di, Mez, and My. So then the only way that you can really make a nice sentence out of it is to say, well, could you tell Di that Mez meant My? And if you put that sentence together, you will remember the divisions of the embryologic development of this brain. Now let's talk about what develops from these guys. So you already have a skeleton of knowledge to know that tel, die, mes, met, my. So you know that tel is at the very front of the nervous system. So what would that create in the brain of the adult? Well, it makes sense that it would make the cerebral hemispheres. What is the next structure down from the cerebral hemispheres? The next thing that is connected to the hemispheres is the thalamus. Okay? When we talk about the sensory nervous system and where sensory information synapses before getting to the cortex, you'll see that there are a lot of stops in the thalamus. So it makes sense that the very last stopping point before the cerebral hemispheres is the thalamus. And that's another way to try to commit this to memory. So what is the mesencephalon? What is the mesencephalon going to relate to in the adult? Well, again, the next stop right before the thalamus on the way up to the brain is the midbrain. So that's what the mesencephalon is forming. You're going to remember this well because in the next section where we talk about the brainstem, we're going to talk about the divisions of the brainstem, and you will know it cold. So you'll know that the next section down from the midbrain is pons and cerebellum. So that's what the metencephalon will form. And then the final stop when you're leaving the brain is the medulla. So I wouldn't say it's a stop necessarily, but it is a conduit for a lot of the fiber tracts. So tel, die, mes, met, my translates into cerebral hemispheres, thalamus, midbrain, pons, medulla. Now, again, you'll notice that I'm sort of trying to build this up as a skeleton of knowledge that we then put different bits of tissue onto, meat, such as muscles and things like that, so that we can build a full body of knowledge for you guys. All right? So now we have to talk about the ventricular system. So what makes sense which part of the ventricular system would be within the cerebral hemispheres? I would say that the lateral ventricles would be there. That makes perfect sense, right? So now let's just walk this down so that we know where the ventricular system is coming from. So the center of the neural tube from the telencephalon is forming the lateral ventricles. What is the ventricular system that is next down from the lateral ventricles? Well, that would be third ventricle. All right? So where does CSF flow from the third ventricle? When it goes through the midbrain, there's a little conduit there. It's called aqueduct of Sylvius. And CSF flows from the aqueduct of Sylvius down into the fourth ventricle. Now, from there, CSF pretty much exits out and bathes the brain, but there is a little central canal that could be in the medulla, which would also be the lumen of the spinal cord, or also known as the central canal. All right, let's look at the gray matter of the spinal cord. What you'll see in this figure is that there is a butterfly of gray matter within the center of the spinal cord. That butterfly is full of cells. So when we say gray matter, that means neurons. If we say white matter, that means axons. So what we're going to talk about right now is the gray matter of the spinal cord. It's divided into the dorsal horn and the ventral horn on both sides. There is an intermediate cord, but the most important thing is to think about the dorsal horn and ventral horn, dorsal being sensory and ventral being motor. how do we form this butterfly of cells? Well, the wall of the neural tube is lined with cells called neuroepithelial cells. You could think of it as the epithelium of the wall of the neural tube. These cells will divide so rapidly we call them neuroblasts. Well, I don't know if that's really why we call them neuroblasts, but you could think of it as almost explosive cell division that's going on during embryology. So they're dividing so quickly, and they're filling up the center of the spinal cord, or what's going to become the spinal cord, with two layers. The dorsal side will be called the alar plate, and the ventral side is aptly named as the basal plate. Well, another way to remember this, other than knowing that basal means ventral, is that the alar portion of the, you have your alar wing of the skull and the mandible, and the alar is up higher, so you could say potentially that that is the back side or the upper side of the spinal cord. Alar, the reason why it's important to remember that is because you're going to need to know that what these things form, what kind of cells. The alar plate forms into the sensory portion of the spinal cord, and the basal plate forms the motor portion of the spinal cord. And that's going to serve you well for a lot of what we're going to talk about in the spinal cord. You need to know that the back side of the spinal cord is important for sensory perception, and the front side, or the ventral side, is responsible for motor output primarily. The separation between these happens to be called the sulcus limitans. Okay? But the important thing is to remember the back side is sensory, front side, or the ventral side, is motor. So now we've covered that. Well, how are we going to make the white matter that surrounds that butterfly of gray matter? And that's called the marginal layer. So that marginal layer will be what surrounds that mantle layer of neuroblasts. So here we have a sort of summary of what we just talked about. The mantle layer forming gray matter, and the marginal layer forming white matter. And remembering again that the dorsal side is sensory, ventral side is motor. So at 3 months of the development, the cord is the entire length of the embryo. Okay? So you can imagine that there's a lot of nervous system growth that is in place even before the body starts to really grow. At the time of birth, the vertebral column growth has way outpaced the growth of the cord. Therefore, the cord actually ends at a level of L3. And as an adult, the cord retracts further within the vertebral column as the vertebrae grow, and then the cord will end at about L2, L1. We say, as neurologists, that myelination is completed at about 1 year of age, at least in the cord, because that's when we start to see the Babinski test, or the plantar response, reverse. And a normal plantar response in an adult is flexor. But if you have demyelinating injury or any sort of central nervous system injury, we'll talk about upper motor neuron and lower motor neuron deficits in the next section, but we see an extensor plantar response if there's damage or demyelination. In the infant, there is no demyelination, it's just that the area has not myelinated yet. So if you scratch the bottom of an infant's foot, the toe will go up as a positive Babinski sign, and that reverses at about 1 year of age. It turns out that myelination in the entire nervous system continues, probably into about 7 years of age even, as a child is developing. But for the purposes of the boards, we would say that myelination in the cord is completed at about 1 year of age. Here's what it would look like. And again, I hope you're staying awake on this. So you want to look at an image like this, and you really want to be able to determine what it is in a short period of time, especially for the USMLE boards. Most people, when you're reviewing, you may be able to just say, oh, well, of course this is going to be something to do with the nervous system. But when you see the board, when you're at the boards, you're not really going to know exactly what section they're going to be testing right now. So you may look at this mass and say, oh my gosh, oh, there's something sticking out of this baby. Is it an omphalocele? Or is it a gastroschisis? The omphalocele comes from the center. Oh no, wait a second. This is the back of the baby. You can see that there's something different there. Oh, so now you know that this fleshy mass is coming out of the back of the baby. Therefore, it must be something like a myelomeningocele, or potentially even just a meningocele. This concludes the embryology section of Falcon Physician Review's Neurology Review.